Okay, welcome everyone. I'll let you grab your last cookie bite, and it's a pleasure. It's a pleasure for me to introduce our speaker today at the Jones Seminar Series. Dr. Han is the Zachary Endowed Professor and Department Chair of Mechanical Engineering at the University of San Antonio. He received his PhD in solid mechanics, biomechanics from Xi'an University in China, with joint training at the University of California in San Diego, under the tutelage of Professor Y.C. Feng. Dr. Han is currently was an associate professor, at Xi'an University and a research engineer at Georgia Institute of Technology before joining the UTSA in 2003. He's obviously had a good career there for 17 years, and is now department chair. Dr. Han's research interests are in the area of cardiovascular biomechanics with a focus on arterial wall stress instability, cardiac mechanics, and tissue remodeling, which he'll tell us about today. He has published over 100 peer-reviewed journal papers. He received a career award from the NSF in 2007, and he's a fellow of the American Heart Association, the College of Fellows of American Institute of Medical and Biomedical Engineering, and the American Society of Mechanical Engineers. So it's a real pleasure to have you here today, Dr. Han, and we look forward to your talk.
Thank you. Thank you, everyone, for coming. It's really my great honor, pleasure to come here to visit you all. Thank you, Brian, for the detailed introduction. I come here actually first time, never been to this part of the country, and although it's cold, but it's really beautiful outside there. And I'm very impressed with all the facility, all the faculty, and this large group of students you have here. It's really give me a lot of opportunity to learn quite a few things. Dr. Jiang showed me your facility of the hospital side as well, so it's really very impressive. So today, I'm trying to share with you two of my research stories, two projects we'll be working on. I know it's maybe a little bit away from your research area, but I hope it can be something interesting, a little bit interest to you, and just see as an example of research project, how it's developed and how it's carried out. I come from UT San Antonio. We are in a mechanical engineering department. So we have a joint PhD program with a very well-known research institute there called Southwest Research Institute. So our PhD is a little bit smaller. We have only 45 students, but we have a large undergraduate population. So it's about 1,200 students. And so our research is a traditional mechanical engineering, so including all the different areas like robotics, thermal fluid, aerospace, manufacturing, and mechanics material, and also people like me doing bio stuff. So we are actually a Hispanic serving institution there. So if you later on need any minority institution to be a collaborator, we are one minority institution there. So I've been very interested in the cardiovascular system since I was in graduate school. So cardiovascular disease is the leading cause of death in the world. So any country now this is the leading one, not the coronavirus. So that's a tiny thing. All right. So I just want to share with you two of the projects we'll be working on. One is related to the artery tortuosity, and one is the heart failure part. So I know many of you engineers, I think many of you took like a solid mechanics or mechanics materials, right? So we talk about the structure, engineering structure. There are always, one is a failure, like a break, right? Damage it. Another type is called stability issue. So things can lose stability under certain load. For example, if you have a long bar, actually I have here examples of rail structures, like the rail during the summer, right? Too hot, so the rail can buckle, loss of stability. So the classic theory is what we call Euler column theory. I think many of you, if you took the solid mechanics, and that's kind of a model you have. So it's a long column under compressive load. So that's the very basic mechanics problem. And this long bar can lose stability, what we call buckling, if you keep increase the load. And that is the buckle into a wave shape. And that's a classic mechanics theory. And there is an equation called Euler equation. You can actually predict what load it will buckle. So that basically is a structure under compression, like this microphone here, right? If you keep compress it, it will buckle. I'm not that strong, but I'm strong enough to press on this will buckle, right? So that's the kind of phenomenon. So that's can be under compression. But if you look at the arteries, because I'm interested in the cardiovascular system, so look at the arteries. If you squeeze it from outside, it can collapse. That kind of loss stability, right? But if you look at the artery, actually in the body, it's under high pressure and also under excitation. We know that because when you cut the artery, it will shrink back to a shorter length, right? So it's under excitation and the internal pressure. So we think that should be stable, right? And we always think that's stable, right? Because it's the tension. However, maybe it's not true, okay? The reason is, so when I was, okay, so when I was in the lab running some experiment before, so what we did was perfuse a segment of artery. We tried to run some biological experiment on this artery. So basically I stretch it, right, try to recover the wavelength, and then perfuse it with the pressure. So when you increase the pressure here, for example, you see here under 100 million mercury, that's a normal range. So the vessel is supposed to just inflate, right? Under tension and inflate, that's fine. But one day when we ran the experiment, accident happens. So my flow loop was blocked. So the flow cannot go through smoothly, and the pressure build up. And this is what happens. So the vessel just suddenly went to the side. So what's happening? I don't know. It bothered me for many times, actually many years. I don't know what's going on there. And this actually, I couldn't explain because from mechanics, what I learned in graduate school was when you stretch in one direction, well, you shrink the other direction. Now it's like inflate. Then it's supposed to be pulling there and it's supposed to be stable. Otherwise, it's buckle. So this problem bothered me for many years. I kind of push it aside. Okay, don't pay much attention. And then when I become a professor at UT San Antonio, so I say, we're looking for a project to try to, develop a proposal to work on. So then I just somehow find my own notes there. This may be a problem. So eventually I think, okay, let's say you suppose not buckle, not bend, right? So why it's bend? So now one day suddenly I got after spring break, right, with no answer. But then the next week, somehow one day I feel, what if I assume it bent? Can I produce that condition, make it bend, maintain that condition? And that actually is the typical stability theory. It gives a small disturbance, whether things can stay there. So basically, what I did was that. So for example, from this cartoon, you can pressure there inside the vessel. Now imagine you have the pressure hard enough, and the vessel just buckle, bend over, right? See if you can predict what's the condition that will occur. Then you know it can happen. So with that in mind, then I look at this, maybe I should stay from there. So then I just look at, so we look at the artery. Let's say if it bend to the side a little bit, then if you look at the small segment of this vessel, so what happens is one side, like an accordion, right? One side you stretch it, another side you compress on it. So the area will be different. Now, if you have the same pressure there, then the force compressed on the bottom and the top will be different, right? So that, if you look at the whole beam there, like you have a distributed load to just push to the side, although you just have a pressure. So with that, and then you can actually now look at the differential equation or geometry, do the analysis. You can actually figure out what's that load level. And then with the classical mechanical theory, okay, if you're interested, you can go look at the paper. So I won't go to the detail. But with the mechanics theory, you can set up the equation of equilibrium, the classic mechanical differential equations. And then you can solve it. Well, you say, no, there's too many unknowns, right? You don't know the pressure and you don't know how it deforms. But it's a classical, what we call eigenvalue problem. That's the mathematical term. So you can solve two unknowns with this one equation. And basically, we solve it, and you can find that's the equation. So now I have an equation. Oh, this pressure should buckle. So then you can do theoretical calculations, right? And then we can do experimental tests. So basically, that's what we did. So here is the experimentally, we measure, get a piece of artery, right? You do the buckling experiment, and then you measure the mechanical property, and then you can build to your model, do all these calculations, right? So, and then you can compare to your experiment. Because of that, we can test it. You increase the pressure and say, measure at what pressure, oh, this occurred, and then you record that. So here's the result of 4 of the vessels. We did many, but here are the 4 vessels. You can see some predicted very well, some not so well, but in overall, seems that occurred. So experimentally, we say the phenomena, and theoretically, we can prove it's there. Of course, you can also do computer simulations to see that phenomenon, and we did that later on as well. So basically, this says, yes, a vessel under tension and the internal pressure can buckle, lose stability, and generate this shape. So we kind of, at the beginning, we tried to publish a paper, got the several rejections, but eventually we got to publish. People finally accept it this way. So with the first paper out, then the second, the third will be much easier. So people kind of believe you now. So then what we did was, okay, in the vessel, arteries are under pasta flow, right? So what I just showed was just assume a steady pressure. So what happened in the pasta flow condition? So this what we did will be we set up a flow loop, and then you can provide the perfusion flow to the system. So you can adjust the wave, make it like simulate the blood flow. And so this is what we see here. So this is a video. Let me see if I can play it. Yeah, so you can see with the pasta, the vessel just bending. So it's more like a dynamic vibration, right? So that's what happened to the vessel. But it's basically, this says dynamically, it also occurs. So it don't have to be static pressure, right? So that's what happened. Okay, so with that, we kind of say, oh, yeah, artery can buckle, and we can actually have equation some way predicting, estimate the values. So then the question back then was, oh, so what? It sounds interesting phenomenon, but who cares? Why it's important? So we look at the blood vessel in the body. Actually, we found there's something related to this, although we were not sure at that time, and that's what we see in the blood vessels. So lots of time you see very tortuous or curved or bending or twisted. People use different terms just describe this kind of phenomenon. So the vessel can become really tortuous in shape. We say, oh, that's tortuous shape, but what we see here is that. So then the question was, is this possibly the buckling is a mechanism for this? So that's basically our research question. Okay, so actually I have one more slide to show you this occurring a lot. So this actually, when I went to Texas A&M, talk about this phenomenon. So one of my friend there, he gave me his image when he was a graduate student. So that's his arteries, okay? And then he is a professor at A&M, so he gave me his current vessel. So that's when he become a professor, that's what his vessel look like. So with aging or with some hypertension or disease, so basically his blood vessel become really tortuous. So actually I know quite a few people, they have this kind of curved vessels, right? So then we say, oh, is this possibly because of the buckling thing or maybe not? So we try to say if there are any link there. Another phenomenon is the veins. In the summer, you say some people has a varicose vein, a spider vein, they call it, right? So, during the summer, you say So maybe the vein have this shape, maybe also can be a mechanism. So that basically triggers, oh, maybe there's a mechanism, so we want to do a further study whether that's linked or not. So clinically, there are papers about this spider vein or the curved vessel. So they more look at the correlations. Say hypertension may relate to it, or aging, or the other factor related to it. So but we want to say if buckling can be a mechanism. Let me say here. Okay, so then we asked this research question, as I just said. So is this a linked mechanism? First thing we did was, okay, we know the artery will buckle. That's what I showed there. But then in the body, you see, oh, it's waving. It's not just one big wave there, right? Why is this one big wave when sometimes it's multiple waves? So one thing came to mind was the vessel in the body, there's surrounding tissue there. So we want to first say, if you have a surrounding tissue, how would the things buckle, if buckle is a mechanism? So we look at it very simply. So I had an undergraduate student working in the lab. So he just go to Walmart, brought some Jell-O. Okay, so then we solidify the Jell-O and then make it like a surrounding tissue, mimic. It's not tissue, right? So basically, we have this Jell-O around this vessel. And then you do the same experiment. This is actually the same vessel before and after. So you see now the vessel buckle into multiple. So that basically say the surrounding tissue play a role. You know, from mechanics, you know the elastic foundation, the classic Tiomchenko theory, the elastic foundation will play a role in the deformation of this structure. So basically with that, then we go back to the mathematical drawing and the mathematical equations. So we actually can assume there is a tissue surrounding the vessel, and then we can similarly, like the differential equation I showed, you can get this solution. So one interesting thing here, you see the N, right? The second term and third term. So that N is the wave number, how many waves you have. And you can see here, one N is at the top, N squared, one is the bottom. So that means you have a minimum value. So if you plot that curve, actually it's the one in the bottom here. So you see at some point, depend on the other parameters. So you may have a N equals to 3 is minimum, or N equals 4 minimum. So that means you may buckle into 3 waves or 4 waves, take the least energy. So the vessel will deform to that way, right? So basically now the vessel will buckle into multiple waves, depend on the environment. And that will be kind of showing, really to what you see in the body, multiple waves. But this is still not a solid evidence, right? Correlation, still kind of correlation. So we want to do further. So there are a couple of things we did later on. One was we look at the growth of the vessel. So if you have an artery, right? Oftentimes the main branch get blocked, but the artery has network. So then the side branch will grow a lot. When it grows, it will become really wavy. So we model that and find actually the buckling pressure decrease. You can see my postdoc, Qing Liu, he did it. So basically you see a reduction of this pressure required to generate the phenomenon. So in other words, when the tissue grows, it much easier will become curved. So that's one. And this actually growth lead to kind of buckling, waving shape. It's pretty common in lots of the plants. So here I collect a few pictures of the beautiful flowers and the fruit. So there are people studying this kind of phenomenon. So they actually show these patterns, all these flower patterns are due to non-uniform growth, right? For example, if you go home, you tear a garbage bag. When you tear it, you can see the edge, it will be waving shape. It's like this happening there. So basically, buckling generates shape change. It's common in the plants or in other tissue. Okay, so another thing we look at this link is for aging. So with aging, as I said, that professor friend of mine, he has this tortuous vessel, and more data showing with aging, the vessel often become curved. Right? And so with aging, there are two things happening. One is the vessel gets relaxed, so the tension in the vessel is not that strong anymore when we're getting old. And second, the component of the vessel change. There is one component called elastin. It's important for the mechanical property of the artery. And that part gets degraded. And we lost some of the elastin with aging. So there's two factors we look at it, right? And in another situation, this is the one, a family of 4, they have a very tortuous vessel. There's not aging, that's because the father, mother, son, daughter, 4 people, so they different age. But they have a common thing, they are short of one gene defect. That one gene defect causes the elastin degradation in their vessels. So they show a very tortuous vessel. So we want to look at the axial tension, we know for sure, yes, when you release it, right, the tension you will buckle. And then we look at, okay, what about elastin? That's compounding chain. So we did get some blood vessels, and then we artificially put a compounding elastin, and we try to break down the elastin. Right? So this way you break down artificially, right? You break it down, mimic what's happening in the disease condition. And this the result we get after you break down. So you can see a significant drop in this critical pressure level. So when you have a breakdown, it's much easier. You need a little bit of pressure, you can buckle the vessel. So the vessel can become curved. Right? So that's another thing. So basically, those few things combined together, we think, oh, this is a link together, right? So we will actually next will try to experimentally observe whether buckling can lead to the tortuous vessel. But before that, I just want to show quickly, or maybe I can jump to this one. So we just say, okay, what happened after the vessel buckle, the curved vessel? So the curved vessel we see here, when you have the blood flow through, so it's easy to generate thrombosis. That's some research show, not us, but the people show. So we did a simulation, and we show the similar phenomenon. So here is a simulation. You know, all the particles mimic the thrombosis process, right? So we have the platelet go through the vessel, and you can see accumulation, deposition of the platelet around the wall at the corners. And this match the experimental observation from other groups. So this top is the experimental observation. And so this is like easy to understand, like a river, curved river, right? The sand silt will basically form around the river. So that's from fluid side, that's it. So next, basically, we just want to say, if we have a buckled artery, whether we can lead to a curved vessel. So some experimental observation or measurement. So we did 2 experiments. One is we have this flow system. So I put the vessel in the system, and then we can just basically increase the pressure, generate buckling. And then you see how the vessel will adapt to that condition. So what we did was, for example, we measured how the cell proliferate, how the cell grow, how the matrix change. Right? Another one is we developed an animal model. Actually, Dr. Zhang, he is a heart surgeon. So he came here a couple of years ago. So he did all this experiment for us. So basically, get a piece of one side of the artery of a rat, and then you cut a segment, right, and then move to the other side. Transfer to the other side. This way you create, as you show here, create a loose connection, and that vessel will buckle under the pressure. And then you want to see how this vessel adapts. So we basically look at this and say what's happening here. And so our result basically tell us, yeah, once you do this, once the buckle, it will stimulate growth and stimulate now uniform growth. So the 2 sides will grow differently. So if you keep growing differently, imagine if you can do a longer term, right? It will be permanently. Okay, but with this system, we can only do 1 week. So we did then for the animal model, we did 1 month and showing the similar thing. So the growth is increased. So with that, we kind of say, yeah, the buckling can lead to this tortuous shape. All right, so that's basically the main part of the story. But afterward, since we're doing buckling, so, you know, computer, you can simulate, or experiment, you can try different things. So we actually also tried like we call twist buckling. So we twist the vessel, what happens, right? So you can apply all different load. So we basically get the vessel and just try to twist it. And this is what happened as well. So when you twist it, initially it's just still a tube. And a certain load, suddenly there is what we call a kink occur in the middle there. So if you look at the vessel, there's a narrowing in the middle. So that's the kind of a twist, right? So then one was interested in computer simulation. So he actually simulated all kind of different weird shape can occur when you're applying different load. So this is basically the artery buckling study here. So I don't know if anyone has a quick question. We still have time. So next I want to show another recent project we're working on. But any quick questions? No? Okay, all right. I'll just. Yeah. That's our basic hypothesis. Yeah, so we show some evidence prove it, but I don't think we completely 100% nailed down that because we don't have data, say 3 months, 6 months, or a year, or 10 years, what happens. We don't have that data. So we need to work on that part on hard. But 1 year study is not easy. At least right now, we don't have the fund to do much longer term yet. Any other questions? Okay, so next, actually, I have this slide, just quick conclusion of this part. So the artery can buckle, as I showed, you'll buckle into this waving shape, right? If the pressure is high. And this can be a possibly affect the flow, and then vessel will grow, adapt, and that eventually may be lead to tortuous vessel. Okay. All right, so next I want to share with you a recent story that project we've been working on, and that is slightly different. It's on the heart. So we look at the heart failure. Say if we can develop a new treatment. So the reason is, right now we know a lot. Most of the time we know about someone has a heart attack, right? So the muscle cannot contract, and that's heart can fail, and that is we call actually a systolic failure. So the heart cannot, as a pump, cannot beat. Right? But there is actually more common, more patient is the other type. It's called diastolic heart failure. That's with the aging population with hypertension, long-term hypertension. So the heart gradually grow and become thicker. The wall become thicker and stiff. So stiffer than normal. And so the heart cannot relax good enough to get the blood into the heart and then pump out. So no blood there to pump. And that's can lead to another type of heart failure. And unfortunately, this one, there is no effective treatment. The other one you can do bypass or you put a stent. So there are some treatment effective immediately, actually. So this one don't have effective treatment. And so we will say, if there any way we can develop a surgical technique to treat it. So that's the basic idea. Because we see here, as I show this picture, so the airway, the growth, hypertrophy, you can see the wall thickness is really big and normal. And also all this thing grow, right, inside, outside, so they all grow there. So when we look at the heart, there are a special structure component in there called trabeculae. So these are the small muscle bundles. There's a lot in the human heart. And this is actually a unique feature for human heart. We look at the pig, we look at the sheep, look at the rat, they don't have much of this trabeculae in there. But the human has a lot. Right? And this trabeculae, the small muscle bundles, most of the time people just ignore it. They don't think it's play any role, any use. But it actually change with the heart. So when the heart gets stiffer, the trabeculae are also get stiffer, stronger. Right? So then the idea we had was, let's say if we can somehow cut the trabeculae, and then you can weaken the wall. So the general idea is because the wall becomes too stiff, right? You want some way weaken, but you don't want to cut the wall. The reason is this, all the cardiac surgeon, they concerned about if you cut it or break a leaking heart, then fail, right? So basically, no one wants to cut the wall. It's not considered not safe. But if we cut just the inside part, maybe it's safe enough. So that's the basic idea, right? But when you do the cutting, what would happen? Can it really change the compliance, right, or the stiffness of the wall? Okay. So that's the first question we have to answer. And so what we did was we experimentally tested if you cut it, what will happen to the heart, how it affect its stiffness or compliance. So what we did was get a group of human heart explant. So you cannot do live experiment in any person, right? But there's explant heart. There's no use from the from hospital. So we collect that heart and then put it in the system, try to give a pressure, right? Inflate it, and then see how stiff they are. So you can measure it. And then cut it, and then measure it again. Then you see whether there's any effect. So here, a group of 6 patients we did. So you can see the pressure volume relation. That kind of reflects how stiff the heart as a chamber, right? So you can see here, the solid dots are the one before with trabecular. So the hollow ones after the cut. So you can see kind of a relaxation of the stiffness of the heart. But this is just, dead heart. You just try passively, right? You don't know the function. So then the next question is, okay, how it affected the contraction, the beating part of the heart? It's because if you want to do something in the human heart, that's a functional beating heart. So we cannot do this experiment in the human. So the first idea we came up with, let's just do finite element model, computational models. As mechanical engineer, we do all the finite element models, right? Numerically, you can test it. So what we did was get a human heart, use the MR image, you can scan human heart, and then you can get the structure. And it's actually real structure, right? So it's patient-specific, detailed structure. And then with that image, you can reconstruct the heart model. And then with this heart, you can artificially do a couple of things. One is, for example, you can say, let's say we cut this part. So then you just take this part of the material away. And then I'll cut here, so I'll take this away. So you can build different models, kind of mimic the cutting process. So we actually compare 3 types. One is we just cut a portion of the trabecular, like the pulmonary muscle, everything will stay there. Another one is say, let's cut everything. Virtually, you can do that, but in reality, although you cannot do it, right? So, but anyway, computer models we just did. So we basically developed this 3 model based on the same patient image information. So then we can put in the mechanics, right? Your muscle, fiber, direction. It's complicated, but there are information there you can use. Mechanical behave, there's other equations, you can put it there. So I'll put it there. And then you mimic, okay, there is a pressure and let's say the contraction. So we actually use a software called FE Bio. And that actually can simulate the contraction part as well as the relaxation part. So if you want to see, actually, here's the equations. I don't think we have time to go all these details. But basically, there are set equations. You can mimic the contraction part. If you're interested, you can read the paper, find the detail. So you basically can simulate contraction process and the relaxation process. So that's what we did. And then, for example, here is one heart with everything there. So I think this is a video. So you can see we can mimic this contraction process, right? And this for intact. And then you can simulate another model without this trabecular or without anything. So then you can compare how it affects. So that's what we did. And here's basically the result. Okay. So you can see with different level of cutting, you have different pressure volume relation. That's the passive part. And then this part is the systolic. It's the contraction part. And that's kind of the the function curve. Clinically, they use this PV curve to show how the heart is behaving. Right? So we use the same thing to compare how they behave. So that's actually showing if you cut it, right, you can see here it's changing the function, improve the compliance, right, and without significantly affect the systolic function. The systolic function is more the slope. The slope will change much there. So that's the model simulation kind of tell us But that's just a model, right? What happened in the real heart. So the next thing we came up with, we cannot do a human experiment, but we can use animal model. So as I said, the pig won't work. The pig no heart, no trabecular. So we find actually New Zealand rabbit. Many other rabbit don't have, but the New Zealand rabbit has a lot of trabecular. So we actually get some New Zealand white rabbit, and then we get this heart, right? And then take it out to a perfusion system. So you provide the nutrition, so the heart will continue beating outside the body. So we call it ex vivo kind of condition. So the heart still functioning. And this way, actually, I think this is the video. You can see the heart beating there, right? So the heart can beating many hours, and that give you a window. You can do experiment. Test the function, and then you can cut it, and then test it again. So the first group of study we did was have 3 sets of the animal rabbit heart, right? One you cut a little bit, one you cut a lot, and then one you don't cut. So you have 3 groups you can compare. Right? So, and then this actually, if you look at the pressure volume relation, that's what you see here. So systolically, the function seems not change. So basically, the beating part not changing much. Right? And diastolically, the relaxation, you can see the stiffness is changing. So it's improved. It's less stiff. You can see here. The red one is the more aggressive cutting. You cut it a lot. So you can see the compliance is reducing. Right? So basically, that's what we're looking for. We want it to get more compliant, more less stiff, so it can relax, but don't affect the beating function. Right? But these are the 3 different groups of rabbit hearts. So if you plot the compliance, you can see here, right? With the level of cutting, you have more compliant heart. So if you want more compliant, you cut more. So that's basically it. But these are different hearts, so it's not a for medical study. Many of you do the biomedical study, so you need the control. Right? So this control is different set of heart. So then the question will be, what if you do the same heart? So then we add the second group of experiment, and that was we try to test it before and after in the same heart. So gradually, with the technique improve, we can do that. So this is the one set of experiment. So you can see the change of the function before and after of the same heart. So there, and you can still see here a significant change of the compliance. So this part occurred. So here the star showing that difference, right? And systolic function, we do see some change, but it's not that significant. So it may affect the systolic function slightly, but it won't be significant. That's the conclusion. So here actually some of the data we have here. Yeah, so some of the data that evaluate the heart, that's a standard. People study physiology, they measure all this kind of parameters, so we did the same. So for example, one interesting thing here is if you look at the bottom, this capital bodied line, so basically we see a compliance improvement significant with a significance. And a little bit surprise thing here is the second line, the top there. So the systolic function somehow actually improved that slightly, not significant, but improved a little bit. So we're not sure why, but that's what the data showing. So anyway, I think the conclusion is that we can actually cut the trabeculae that improve the compliance and without significantly affect the systolic function. So this is ongoing work. Right now, we're still trying to develop the procedure. Say next step will be we want to get a better model, like more detailed heart structure with more detailed trabecular information. And also we try to develop the procedure. How you're going to translate this to human. Right? You want some minimum invasive procedure you can do, but how you're going to put the knife in and see the trabeculae where you cut in. So there are lots of technical challenges. Many problems need to be solved there. So anyway, we're still working on this right now. All right. So with that, I think that's basically the study on the heart. If you are interested, you can read the paper. We have a couple of papers just came out 2019. Well, with that work, actually, I have to tell you, lots of people did this work. There's two simple story, but they need a lot of manpower to do it. So this first group of people, they work on the artery buckling project. Many of my students, postdocs, most of them already left my lab. But they did mainly the artery buckling study. And then the second group of people, we did work together, did the heart. So actually, this one have Dr. Mark Feldman. So he's my collaborator there. Actually, he, the first one, came up with this question. He asked, "Oh, can we do a cutting, to improve the function?" Yeah, since we are mechanics person, so they, "Oh, can you do the modeling simulation so we can kind of optimize the plan, how to cut it or mimic it?" So basically, that's why we work together at this time. Actually, Dr. Zhang here collaborates with Mark Feldman as well, so I met him through Dr. Mark Feldman. So anyway, this is teamwork. Lots of people contributed. And this was also supported by this one by the American Heart Association. And now we try to get some NIH funding. Hope we can get it and continue to do this research. The artery buckling project was supported by the NSF and the NIH. That's actually my career award proposal on the artery buckling. That's you just mentioned. Okay, with that, I want to leave. We have some time for questions, but I want to say thank you all for coming. Thank you for your attention. So this slide is San Antonio, the Alamo. So hope you got a chance to visit San Antonio. That's downtown. There's a river walk. It's a beautiful city. It's hot in the summer, but the winter is warm. All right, thank you.